patterns of genetic inheritance. And we studied very similar chapter, patterns of chromosomal inheritance. What is the difference? So remember that chromosome is a DNA that is wrapped around proteins and then compacted again and again. And if you lose one chromosome, it's just like losing the whole truckload of genetic information or acquiring a truckload of genetic information that will mess up a lot of processes at once. And it's Down syndrome or poly-X syndromes. Such changes are easily detectable by looking at the karyotype, which is a picture of your chromosomes. So it's basically the losing or getting a big chunk of genetic information. What we are going to study today is the interaction between alleles of the same gene. Okay, once again, I'm gonna get some new vocabulary. We're going to be looking at the same gene. What is a gene? It's a piece of DNA, Gattaca, ACT for a long, long time, which codes for one protein. Like let's say eye color is a gene for simplicity. How many copies of the gene have you got? 2. Yes, because you get one copy of the gene from your mom, you got the other copy from your dad. And that's why every gene is repeated, every gene is repeated once in our genome, but it always comes in 2 copies. That, the chromosomal set, basically the same chromosome, the same type of chromosome, one came from your dad, the other one came from your mom. The question now for this lecture, what to do if those alleles are slightly different? Like for example, from dad you got blue eyes, from your mom you got brown eyes. What the baby gets? So this is the interaction between alleles, and this is related to learning patterns of genetic inheritance. Let's look at our vocabulary that starts with the word allele. And this is a gene version. So never be confused if you're talking about 2 genes or 2 alleles. If you are talking about 2 genes, you're talking about 2 different proteins, 2 different places on the chromosome or chromosomes, 2 genes. And each of those genes come in 2 alleles, 2 versions of the gene. It's very easy to be confused. Are you talking about genes or allele? Because allele is a gene, but it's the word allele is reserved for gene version. And you can have only 2 versions of the gene. Here's an example. But in human genomes, some genes exist in many versions, like blood types. You have blood type O, blood type A, B, or AB. We're going to be looking at that in details, but in all of these cases, we're talking about the same, the same gene. The gene that sits on the plasma membrane of the blood cells and is a certain receptor, is a certain marker, receptor, marker, something that makes your blood cells specific. And in this case, one allele is called A. It's a certain shape of the protein, let's say like that. There is a certain allele that is called B. Let's say it's marker, receptor, but of slightly different shape, slightly different version. And a combination of this will either give you a blood type of AB, when both of them are present. The blood type A is when only type A version is present on plasma membrane. 0 or O, none of them are present because they are both mutated. In any way, the phenotype in this particular case, which is your blood type is your phenotype. It's like the way you behave, the way you look like. So phenotype is a reflection of genotype. So a blood type is a phenotype, and your genotype is those two allelic versions of the same gene. Okay, it's kind of confusing right now. Hopefully it will become a little bit more clear later on. Now the next word is dominant. Dominant and recessive. They are opposite words, and they refer to alleles. Okay, let me help you. You can hear dominant gene, recessive gene, but in most cases, unless it's polygenic inheritance or more rare cases of genetic interaction, it refers to alleles. Like dominant allele of the gene or recessive allele of the gene. Dominant is strong version. It means that you're gonna see the phenotype for this version no matter what. And recessive is a weak version of the gene. Let's say that the allele that the baby got from its mother is a sick, mutated, recessive version. But if it has a good allele that can make up for the missed work of the bad allele, so the baby is going to be healthy. It's gonna carry this invisible now genetic trait of the disease, but the phenotype of the baby is going to be okay. Because every gene comes in two alleles, and if a good allele, we're not saying right now dominant or recessive, because diseases could be both recessive or dominant. Sometimes dominant version is better. So in some cases you do not see the genotype because non-mutated or wild allele makes up the work of diseased allele, so the phenotype is just going to be okay. But we're going to talk about that a little bit later on. I need a couple of more words from your vocabulary. Now, if we are going to communicate some genetic material, especially if you're a genetic consultant and you have to talk about that to the customer. You have to write down or invent some, writing for the genotypes. And let's say we're gonna talk about the gene A. We are going to, the dominant allele of the gene A is always written as a capital A. And recessive alleles are always written as a lowercase a. So if we have two alleles in the genotype, in every genotype, the combinations would be like that, right? Gene A, and you always have to mark the alleles for this gene, 2 letters. And the size of the letter defines is the allele, if the allele is dominant or recessive. Now here, genotypes of baby or zygote or something. When genotypes, when a cell goes through reproductive system and create egg and sperm. In this case, it can, let's say it's egg. It can create 2 types of egg. In this case, it can create only 1 type of egg, and here as well. And then it's gonna meet sperm, and once again, it's gonna create a genotype with 2 alleles of the same gene. Okay, we have a name for this kind of genotypes. This one is called heterozygous. So zygote is the first cell of a new baby. If you see different letters, different in the sense that different sized letters, one is capital, the other one is lowercase, we call it heterozygous. These 2 genotypes are called homo. Homo means same. Hetero means different. Homozygous. And this one is called homozygous dominant genotype, and this one is called homozygous recessive. Now you have homozygous and heterozygous words in your vocabulary. So how can we define it so you can remember?
Homozygous they're both either strong or weak.
Yes, both alleles are either both dominant or both recessive. This is homozygous. It's going to be important. And heterozygous alleles are different. One is dominant and the other one is recessive. The other kind of vocabulary I'll need is autosome or sex-linked. Remember that a person has 23 sets of chromosomes. These chromosomes, 22 sets, are autosomes, and this one are sex chromosomes, and it's X and Y. So, autosomal or sex-linked autosomal means the location of the gene that we will be talking about. If we are talking about autosomal inheritance, what we mean is that the gene in question is located on one of these 22 chromosomes, which are not sex chromosomes. If we are talking about sex-linked inheritance, it means that the gene that defines the phenotype is located on X or Y chromosome, mostly X because Y doesn't carry a lot of genes. And you will see quite different pattern of inheritance depending on the position of the gene in question, either on autosomes or sex chromosomes. Still confusing, I know. We keep plowing. Let's say, do you know such disease as hemophilia? In genetic terms, it's sex-linked recessive disease. And in this, like 2-3 words, we can say that if I say it's sex-linked, the gene for hemophilia is located on X chromosome. And if we say it's recessive, it means that we need two recessive, this gene represented by the recessive allele. So it means that recessive allele is mutated. Okay, still confusing, I know. Right now, okay, I can't really say look at your ears, but we're going to talk about such personal features as ears. This is unattached earlobes or attached earlobes. Then the hairline, which represents the interaction between alleles. What you see written in this picture is a genotype that produces a certain phenotype. First of all, we are going to look at one trait crosses, which means that we are going to, let's say, look at eye color, gene of eye color, and see how alleles, let's say blue eye color and brown eye color, can interact in the zygote or baby. And just to complete this thing, let's say you are writing a genotype for 2 genes. Different genes are always represented by different letters of the alphabet. This is a genotype, and we can say that if we're gonna be talking about gene A, the genotype is heterozygous. Do you see heterozygous? And if we are going to talk about gene B, the genotype is homozygous and dominant. So if you're saying the word homozygous, you always have to add homozygous recessive or homozygous dominant. First pattern that we are going to discuss today is a complete dominance pattern of inheritance. Complete dominance. If you hear that disease is inherited by the complete dominance pattern, it means that the dominant allele totally masks the work of the recessive allele. You don't see recessive allele at all. So recessive allele in combination with dominant becomes invisible. Okay, blue eyes, here's an example. Eye color, gene. Eye color. So blue, recessive. It was found out that the blue color is provided by recessive allele. Brown color, once again, it's a grossly simplified case, dominant allele. So let's say I'm gonna write you genotypes, and you're gonna tell me the eye color. Let's call it gene B. What eye color is here? Oh, wait a minute. What eye color is here? Brown. Eye color here. Eye color here. Brown. Okay. Now let's say we have two brown-eyed parents, and by the measure of luck, what did I write? Male or female? Male. Male. Meets. This is a sign for meeting and reproducing. So brown, brown, brown-eyed man meets brown-eyed woman and reproduces. With that. We are going to calculate the probabilities of different genotypes. We use a special tool that is called Punnett square. And I'm going to ask you to write a kind of a square. What you write here, let's say male and female. We are going to list gametes. What are gametes? Egg or sperm. For female here, and gametes for male here. Remember, gametes is egg or sperm. So I'm drawing egg right here, and I'm much smaller. Sperm for male. So we won't forget what we are writing here. And we're going to write genotype of gametes. How genotype of gametes is different from genotype of zygote? Okay, the cell goes through reproduction. Huh?
The zygote has both.
Huh?
The zygote has both.
Yes, zygote has both alleles. gametes, egg or sperm, have only one. Okay, and if here's a female, it can produce an egg with dominant allele, but just one. Remember that for gametes, that is the number one thing to remember. Gametes has only one allele. Or it can produce an egg carrying the recessive allele, this one. And the same thing goes for a male. It can produce a sperm of this one, and it can produce a sperm with carrying the recessive allele. Now, these squares are for writing the genotypes of a zygote. When this egg meets this sperm, what will be the genotype of a zygote? That's right. You're good. If you learn how to do Punnett squares, that is basically you pass this chapter. Okay, and this one meets this egg. I will complete the thing. So the first step, writing the genotypes of gametes. Writing the genotypes of a zygote. This last step, assigning the phenotype. Eye color? What is that? Brown. Eye color? Brown. Okay, brown here. What do we get here? Blue. Do you see milkmaid case right here? And two brown-eyed parents have a blue-eyed child. This is possible with complete dominance pattern of inheritance. That's how it could be explained how two healthy parents can produce deadly sick child. Because in this case, we've been discussing eye color. The recessive gene can carry a disease, like for example, hemophilia. If the genotype of a person is heterozygous, you never see hemophilia or some other disease unless this person reproduces with the other heterozygous parent, and with the probability, by the way, what is the probability of having blue-eyed child right here? 1/4. Okay, and with a probability of 25% of 1/4, two healthy heterozygous parents can get a sick child with different eye color. Dark hair and blonde hair. For simplicity, once again, complete dominance pattern of inheritance. Brown hair, and all of a sudden you see blonde child. Well, in most cases, the inheritance pattern, this is classic, but the one trait, complete dominance inheritance pattern is kind of rare. There are always a couple of other genes, not alleles, that can either change the expression of this one or influence it somehow. So we're going to be talking about the other inheritance patterns. But let me give you a few examples where this kind of analysis is totally useful. If you look at your chapter, see Punnett square, this is how to write it. Okay. the autosomal dominant disorders. No, recessive disorders. We just discussed the recessive disorders, and they are all listed right here. Sickle cell disease and cystic fibrosis, Tay-Sachs disease. With dominant disorders, you're gonna see it. You have one copy of mutated gene. You're gonna see it no matter what. Sometimes you're gonna see it at mid-age, but you're gonna see it. Let's go back to our blue and brown eyes and consider a little bit different scenario. Let's say that brown-eyed man with homozygous dominant genotype reproduces with blue-eyed female. And in this case, you know that genotype is homozygous recessive because it's the only genotype that will give you an eye color. So what kind of probabilities of getting a blue-eyed child in this particular case? Huh? 0. Yeah, because the only type of gametes female creates is this one, the only type male creates this. So all kids are going to be brown-eyed. So what probability? Kind of same case, but a little bit different genotype. Is that clear? 50% chance. So remember that genetic works with the probabilities. You can have a disease and your kid can get it with a certain probabilities or not. You may not have, both parents can be healthy, but if there is a lurking recessive genetic disorder, so your kid will get it with a certain probability. By the way, this brown-eyed male is called carrier. What does it carry? It will carry invisible on phenotypically invisible recessive allele. So we call those people who carry invisible phenotypically invisible, let's say hemophilia gene carriers. So this word is widely used in genetic consultant, and it means having a recessive copy, recessive mutated allele, which you don't see phenotypically, which you don't see like showing. Okay, this is part 1, and it refers to autosomal inheritance. With sex-linked, it's a little bit different because sex-linked inheritance always concentrate on certain sex. So if the disease like hemophilia is sex-linked, males are going to be predominantly affected. Let's look at the other patterns of inheritance and then move to sex-linked Punnett square. Okay, for we have incomplete dominance pattern. And I have to change the gene for like uh, let's say it's hair structure gene. Recessive allele will give you straight hair. Dominant allele will give you curly hair. So genotypes, this is going to be curly, homozygous dominant curly, homozygous recessive straight, heterozygous will give you wavy. This is a difference between complete dominance, because complete dominance masks the work of recessive allele completely. Incomplete dominance does not mask the recessive allele completely. And how can you distinguish between those patterns? By looking at the real people. Look at heterozygous. So the definition of incomplete dominance pattern is that heterozygotes have intermediate phenotype. In complete dominance, the pattern was like the phenotype was like dominant allele. In incomplete dominance, heterozygotes have intermediate phenotype. Now another pattern is codominance. You can use Punnett square for codominance, and for incomplete dominance you have to assign the phenotypes a little bit differently than with the pattern of complete dominance. Now, what do I mean by codominance? Blood types. Remember this example. In blood types, there is allele A. There is allele B. And I'm using capital letters because they are dominant. There is an absence of the protein, which means allele O. There is a gene. Here's one gene, one allele, the other allele. The third allele is totally destroyed. It's still there in DNA, but the transcription cannot be done from this allele. The protein is not being made. So this allele is called O, means zero, no receptor protein. Remember, we are talking about receptor protein on the plasma membrane. These two alleles are codominant. In human populations, there are like 3 different alleles for the same gene, but you can have only 2. And these 2 are codominant. But the relationship between A and O or B and O is complete dominance. Now, blood types. Tell me the blood type of a person who has a genotype AO. Blood type A. BO. B. AB. Codominant means the genotype is AB. Both proteins are sitting on plasma membrane. You're gonna see both. It's like proteins belonging to the same gene, 2 different versions of the same protein. You're gonna see both. They're gonna be reacting with both serums. The blood type is AB. you're gonna see the work of both alleles in codominance pattern. Okay? Remember the example of blood type AB, both of them. Now, universal donor, and of course, what is the, yeah, and this is 1, 2, 3, 4, whatever. Now, yeah, it's we still remember why this one is universal acceptor, this one is universal donor, right? Okay, now blood typing by the baby. Mother wants to make sure the old method of finding out the paternity thing. Can a man with blood type AB be a father of the baby with blood type B? Yes, because if a man has AB, it will create a sperm carrying B or A allele. Now the other example, can a man with blood type O be a father of a baby with a blood type AB? Can a man be, by the way, the genotypes with blood type B also. Like that. Can a man with blood type O be a father of the baby with blood type B? Yes, because it can donate this one. And in your homework, there are a couple of exercises of finding out, can a man be father of the baby with that particular blood type? The major thing to remember is that blood type B has 2 corresponding genotypes. This is a phenotype, it's your blood type. These are corresponding genotypes. Okay. There is also, vocabulary. Take a look at your vocabulary. So, complete dominance. The definition is dominant allele totally masks the appearance of recessive allele in heterozygous. So if you have a heterozygote, it's gonna be looking like having 2 copies of dominant. In incomplete dominance, the phenotype of heterozygotes is intermediate. So it's incomplete masking in heterozygote. Codominance, the work of both alleles is shown in phenotype. Remember, blood types A and B. 2 different alleles, you see both, if both are present. Now, polygenic inheritance. Here we are not talking about alleles, we are talking about different set of genes, different set of alleles. So, like your skin, your height depends on the variety of different characteristics. It is controlled by multiple genes. Okay? So polygenic inheritance means that 2 or more genes are influencing phenotype. Once again, what you should remember here is the difference between alleles and genes. Carrier is a person that carries a recessive allele not shown in the phenotype. Just remember that, not shown. You do not see the work of recessive allele in the case of carrier. And trait. Sometimes geneticists like to talk about traits. Trait is your feature, phenotypic feature, like eye color, hair structure. This is a trait. It's a representation of physical representation of the work of particular gene. Okay, the last subject for today is sex-linked inheritance and all this color blindness, hemophilia, and similar disease. Punnett square for sex-linked index. The first thing that you have to remember is that these are two sex chromosomes. So if we talk about some disease being sex-linked, it is positioned on X because Y chromosome is so small, and here's even a theory that it keeps disappearing in the evolution. So it doesn't care about almost any genes except that SRY, sex-determinant region of Y chromosome. So in 99% cases, all the diseases like color blindness, hemophilia are located right here. And the mother is responsible for passing the Yaki disease to her sons, even if the mother looks like normal. Okay, see what happened in this case. Just for you not to be confused with autosomal inheritance, let's mark the defective part of X chromosome with some kind of asterisk or something. It will help us not to forget that hemophilia, for example, is located on X chromosome. So, hemophilia, by the way, what is that?
It's a blood disorder.
It's a blood disorder when the blood cannot clot, it just keep running out, and it creates some, internal bruises, or it's hard to stop the blood. Color blindness. Almost same thing, also the gene is located on X chromosome. Not being able to see certain colors. Now, okay, if the genotype is like that, who is that, male or female? Male. So, colorblind or not? Let's say we're going to discuss color blindness. Colorblind male. Reproduces with normal female. Same thing, you draw Punnett square. Male, female, and what you write here, of course we can use letters here, but I prefer asterisk or something, so it will help you to remember about the disease being located on sex chromosome. And the other one is Y, never forget Y. Then it's XX. What is the proportion of sick kids? Huh? Okay, let's write genotypes. Now what do you say? Sick or healthy?
Sorry.
I forgot to put here recessive. And it's complete dominance pattern for hemophilia and color blindness. So let's try again, sick or healthy? Healthy, but carrier. Male or female? Female. Sick or carrier, healthy. 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 So colorblind male reproducing with a healthy female will give like no colorblind kids. Now let's try different things. Quickly draw another Punnett square and let's look at the probabilities of a carrier female XY. And now here you have to like consider it as like different gametes because it's one egg is carrying a healthy X, the other one is carrying color blind allele. Now let's see what we got in here. Healthy, healthy, healthy. One allele is sick, the other one is healthy, the person is, why don't we write healthy? Sex-linked inheritance is confusing because you have to calculate different probabilities for different sexes. In autosomal inheritance, it's the same probabilities, everybody is equal. In sex-linked inheritance, hell no, no democracy here. Okay, remember, different probabilities. Let's look at the girls. All of them are coming out healthy. Let's look at the boys. Healthy, healthy, healthy. Remember that X does not equal Y. Y cannot complement for X. As soon as at least one copy of the gene, recessive gene, is present, Y cannot contribute to the work of this recessive colorblind gene. This male is sick. So, in this combination, there is 100% chance for girls for being healthy. One is 50% will be carriers, 50% will be will have healthy genotype, but for boys, 50% of them will be colorblind. Who gave them this disease? Always the mother. Always blame the mother. Yeah. Now see, even a different case, hemophiliac female, which is a very, very rare case. See who suffers in this case? With a hemophiliac female. All girls are healthy again, but all of them are carriers now. And all boys are sick. Now all boys are sick. So this is a thing that you have to remember about sex-linked inheritance. The percentage, you have to calculate probabilities differently for males and females. Yes?
So if the mother has a recessive gene for something,
Yes.
then if she has a son, there's probably 100% chance that you'll carry the recessive gene.
Yes. If the mother is sick and visible like that, it's 100% chance. If the mother is a carrier, not a sick person, but just a carrier, there's a 50% chance for boys of getting sick. So boys cannot be carrier Y chromosome does not equal X chromosome. It will not contribute anything. That's why boys are mostly affected. They do not have a protection of a second chromosome. There's also such thing as, sex-linked dominant disease. Where, like, and some of these diseases, if like females will get, some little things are related to that, but I will not complicate our lecture. Already complicated with particular examples. Now, pedigrees. Here is a pedigree, example of the pedigree. And the first thing the genetic counselor does is construct pedigrees. As you can see, the circles are about females, round forms. Males are square, round shoulders, and so on. Okay, that's the thing to remember. Now, the sick individuals have squares or circles colored, and when genetic consultant constructs a pedigree for several generations, by looking at that pedigree, you can tell if the disease is autosomal or sex-linked by just simply looking if both sexes are affected or not. In this pedigree, are both sexes affected? Yes. So the disease is autosomal, meaning the gene is located not on sex chromosome. The second question, is the disease recessive or dominant? How can we tell that by looking at the pedigree? One small rule: if the disease is recessive, it can skip generations. You're gonna do the lab, and you're gonna look at pedigrees. And in your lab manual, if you see the disease skips generation, means parents are healthy, child is sick, this is a recessive. It's carried by the recessive allele, so you do not gonna see it in every generation, kind of lurking in the background. With dominant disease, you're gonna see it in every generation, like in this pedigree. And this is example of autosomal but recessive disease. It just skips generations, but it does affect both sexes. Okay, and that's it for today. Most of your homework is about learning about is about solving genetic problems by using Punnett square for autosomal and sex-linked disease.